Great. Hello, everyone. My name is Dr. Georgios Rigas. I'm a PI of one of the AI for Net Zero projects, specifically in energy and transport, led by Imperial College. And today is really my pleasure to introduce our two early career researchers and speakers of this webinar series on AI for Net Zero. So, as you may know, the webinar series is designed to bring together experts, innovators, and enthusiasts from both the artificial intelligence and sustainability domains, with an overarching goal to accelerate progress towards net zero targets. The series aims to engage and inform communities about the groundbreaking research and developments at the intersection of AI and sustainability. The webinar series will delve into diverse applications of artificial intelligence across various domains crucial to achieving net zero objectives, including energy, transport, environment, agriculture, and food systems. So today we have two speakers with us. The first speaker will be Stephen Gao, and the second one, Timothy. So our first speaker, I'm going to give you a short introduction about his background. Dr. Stephen Gao is a postgraduate researcher in statistics and data science at the University of Southampton. Initially focusing on quantifying uncertainty in computational models, he made a series of sideways moves across the fields of chemistry, demography, machine learning, and data infrastructure. He's now working on statistical methods and data analysis for the Miscanthus AI project. Stephen, welcome on the webinar series. I'm looking forward to your talk.
Thank you. Thank you for having me. Right. Are my slides going to be visible fairly soon? Yes, there they go. Right. I'll click play and then we can go. So hello, everyone. As said, my name's Stephen Gao from University of Southampton. I'm going to be talking about some work that we've been doing for a project that is officially called Plant Selection and Breeding for Net Zero. In practice, we all just refer to it as Miscanthus AI because it focuses on Miscanthus plants. This is a joint project between Aberystwyth University, the University of Lincoln, and the University of Southampton. If you've been following these talks since the start, you'll know that Otar Yeti from Aberystwyth and Ollie Hardy from Lincoln have already spoken about some aspects of this project. So to complete the set of institutions involved, I'll be talking today from Southampton. And in particular, I'm going to be talking about how we estimate genomic effects of and how those influence traits in Miscanthus plants, and how we do that using phenotypic measurements. So to begin, brief overview of what Miscanthus is and why we might care about it. So Miscanthus is a perennial grass which originated in Southeast Asia. It comes in several different varieties, multiple different species and genotypes. It's mostly of interest as an energy crop. It absorbs a lot of carbon, it burns well, it can be used for fuel. It grows quickly, it can reach very large sizes, particularly the species Miscanthus giganticus is particularly effective at that. It grows well in marginal soils and it grows well in a range of different climates. So despite being from Southeast Asia originally, it's relevant in, for example, the United Kingdom, even though our climate is very different. And the plants are relatively low input. Once you've got them growing, they don't need too many resources to keep them going, and they live a long time, so you get a good return from them. And as I mentioned, they can capture a lot of carbon over their lifetime. So the background to this project is that plant breeding is very complicated and very time consuming. It takes a long time to set up a range of different plants. It takes a long time to find out if they've grown well or not. And that's compounded by the fact that what traits you might want in the plant could be different depending on what you're trying to do with it. So a range of different traits could be wanted, and if you have to measure all of them, that's also time consuming and requires a lot of effort. So the overall aim of this project is to try to use machine learning and artificial intelligence to make the whole process of breeding Miscanthus plants with the traits we might want more efficient. And this breaks down into 3 work packages, effectively. There's a work package about data acquisition and analyzing the data that you've collected in the field. This is the one that Ollie Hardy from Lincoln talked about on one of these streams last month. The project I'm going to be talking about today is the part related to machine learning and statistical analysis to select the species and genotypes of Miscanthus that we might want to grow in the first place. And the third aspect of this is sort of related to explanation and argumentation to support decision making. This is particularly the part that involves talking to the plant breeders directly, trying to both make sure that what we discover is communicated to them, and also that we learn from what they've done in the past as well, rather than having to start from scratch. So, little aside here, but it is going to be important later, which traits might we want to focus on? So there are lots of different traits we could care about: how high the plant grows, how wide it is, how resistant to diseases. We're going to mostly focus on 3 for the purposes of this, which are particularly relevant to the energy crop side of things. So we're going to talk about dry matter, which is the total biomass of the plant once the water is removed, approximately. Cellulose, which is the largest combustible component in Miscanthus, and therefore important if you want to burn it for fuel. And lignin, which is also an important combustible component and has uses in energy. So the idea of genomic selection is there are several different species of Miscanthus: Miscanthus giganticus, Miscanthus sinensis, lots of hybrids between them. And within every species, there are potentially thousands of different genotypes. And the idea of genomic selection is to identify which genotype we want to grow, given where the plant is going to be growing, what the environmental conditions in the location are like, and which traits we want to particularly control for, what we want to maximize, what we want to minimize. So you could say, okay, we can just do this exhaustively. We try everything and we see what works. And yeah, we could. That's going to involve growing a lot of plants of different genotypes and effectively averaging across them, because genotypic variation is not the only thing that defines how well an individual plant grows. Different plants within the same species, the same genotype, will still give different results just based on random chance or other factors that are not captured by the genomic variation. This would be, so if we were to do this and to grow everything, average it, this would just be very, very expensive and very time consuming. It just isn't realistic to do this for every possible genotype, and in particular, it isn't possible to do this at every location we might care about. So, what's the alternative? Effectively, the alternative is to try to measure something earlier in the growing process and use this to draw some predictions. So, for example, if we could measure a secondary characteristic of a plant in a non-destructive way, and then just let it keep growing, but we've already got that information very early in the process, so we can already decide what we're going to grow next based on that. And we can use this to kind of predict how well that genotype is going to grow on average. And the main way this has been done in the past is using something called a single nucleotide polymorphism, or SNP. These are a kind of building block of DNA. This image on the right, which I borrowed from Wikimedia Commons because I didn't know how to make one myself, sort of shows what an SNP is. It's simply a change of nucleotide in one base pair location in DNA. So between the top image and the bottom image within this picture, one location has changed from a G to an A, and that is a single nucleotide polymorphism. There is an alternative, which is called near-infrared spectroscopy. I'll talk more about this in a minute, but it's effectively based on how much light a plant absorbs or reflects at different wavelengths. So this kind of leads to a sort of prediction pipeline, where we have the traits that we're interested in on the right. These are the things we want to maximize. Typically, there'll be one or more of interest, and if we have grown plants to measure these, we'll measure them in the examples we looked at 1 to 4 times per plant. So we might measure its height at a particular time, and then at another time, and then again a few times. But if we're measuring dry matter, we'll only measure that once the plant has been chopped down and dried out. Then one step back from that, you have the genotypes, of which there are several hundred or potentially thousands. And in a trial, we might have a few plants of each type. So we can sort of learn how the genotypes behave on average by averaging across the plants and looking at how those affect how the trait measurements differ between different genotypes. But then at the very start of the pipeline, we have these SNPs and these NIRs, and there'll be several thousand measurements of those, which we can take much earlier on. And we need to find a way to sort of see how much of the genotypic variation in the traits can be explained by those initial measurements. The box in green at the bottom, we could also have an extra step between those initial measurements and the genotypes for the species. In the example I'm going to show, we haven't done that. We have one species, but that's another thing you might need to do. So I'm going to talk a little bit about the things that we've measured. So SNPs and NERs are quite different from each other. They behave differently, and we have to take account of this in the analysis. So the single nucleotide polymorphisms are effectively measured on a discrete scale, 3 levels based on presence and absence in a genome. The NERs, near-infrared measurements, these are actually measured on a continuous scale between 0 and 1. And it's effectively the proportion of light that is absorbed at the wavelength. But we measure it across a wide range of wavelengths. In fact, in this occasion, spanning part of the visible light spectrum as well as the near-infrared spectrum. With the SNPs, we could potentially have thousands of these that we can measure, but there are tens or hundreds of thousands in a genome. So the ones that actually drive the genomic variation will generally not be present. We have to assume that we don't have the most important ones, and we need to use the ones the ones we do have measurements for to kind of fill in the gaps. And the nice thing about these SNPs is we can pretty much just throw them straight into our analysis method without too much pre-preparation. We can pretty much just take the levels that they're measured on and use those to build the prediction algorithm. NERs are a bit more complicated. This gets kind of challenging. So to begin with, I'll just show you what NERs look like. So this is a set of spectra from different Miscanthus plants. On the x-axis is wavelength running from 400, so in the visible light spectrum, up to just short of 2500, well in the infrared. And effectively, the plot shows us the proportion of light that is absorbed by each plant at each location. So we can see that around about 500, there's a peak in absorbance where plants absorb a lot of light and reflect a relatively small proportion. Around about 1100 and again at 1300, there are these very low points at which a large majority of the light is reflected rather than absorbed. This image was created by my colleague James Strong from Aberystwyth University, so credit to him because it's a fantastic visualization that certainly made me understand what was going on a lot better. But there's a couple of challenges that come with these, and the first one is that these spectra, each individual spectrum in this plot, tend to be heavily correlated to each other. And we can sort of see that by looking at them. They tend to be high in the same places, they tend to be low in the same places. So this is a plot of the correlation between the spectra from the previous slide. And red is high correlation and yellow is low correlation. So we can see that a lot of spectra, particularly the ones that for plants that were measured around the same time that are perhaps similar in some other way that we've sorted them by, tend to be very, very heavily correlated. The darkest red here is correlations close to 1, which we're saying that we have multiple spectra, but they're basically the same. So this can tend to lead to analysis that doesn't actually tell us very much. A lot of this correlation is kind of artificial. So you can partly improve this by just centering them, just looking at the variation between the spectra with everything set to 0. That's one step. But the way that's really recommended is called a Savitsky-Golay filter. This is a sort of smoothing filter, an idea from pure mathematics, that removes noise from the observations. And this sort of reduces the artificial variation that causes the correlation. And we end up with a plot that looks like the one on the left, where all of those dark red areas are gone. And now we just have lots of spectra that are effectively distinct from each other, and that tells us a lot more about what's actually going on within the plants that allows us to model how the genotypic variation might be related in some way to these spectroscopy results. So the example that I'm going to show that we used this on was from a study called 2TT, which was a set of Miscanthus plants that were grown in Aberystwyth between 2004 and 2009. There were 552 plants, 4 each from 138 different genotypes of the same species. And what this study measured was the data on 20 traits that were of interest, including the 3 that we care about: dry matter, lignin, and cellulose. They also measured SNPs and NERS data for each plant. So the first thing we need to do, if we go right back to the prediction pipeline, is to estimate the genotypic effect, so the effect on the trait of each genotype. The way we do that is using a best linear unbiased predictor. So we take the 4 plants, we sort of average them, but in an intelligent way that accounts for other forms of variation. And what we get back is a sort of a measure of the average deviation from the mean of the plants in each genotype. So if we have 4 plants from genotype 1 and 4 plants from genotype 2, we look at, on average, how genotype 1 performs versus all of the plants in the trial, and how genotype 2 performs versus all of the plants in the trial. And this gives us a kind of baseline estimate from the observed trait data of how each genotype affects each trait. So the way we can then sort of take this forward is using something called a genomic relationship matrix. And the idea here is to define how similar 2 genotypes are based on a set of secondary variables. So we have a set of measurements of the variables that we can measure, which we'll say that's of size M. Those measurements could be SNPs or NIRs. And we have plants from the number of different genotypes, which we'll call N. In this case, N was 138. So we construct a matrix A, which relates the measurements of the SNPs and the NIRs to the genotypes. Again, we do this by averaging across plants in the same genotype. And then we take the cross product of that. So we transpose the matrix, we multiply the transpose by the original matrix. And this effectively gives us a matrix, which is a square matrix of the same size as the number of genotypes that were in the trial. And this effectively measures how closely related each pair of genotypes are based on their SNPs or NIRs. In K, we'll have values that are close to 1. These mean that a pair of genotypes are very similar. So they have effectively the same SNPs present in their genotype. They reflect or absorb approximately the same amount of light at each location along the spectrum. If there are values close to 0, in fact, values below 0 are also possible, thanks to the way this is set up. That means they're very different from each other. So they absorb very different amounts of light. They have none of the same polymorphisms present in their genome. So effectively, now we've built this matrix, which is cool, and we've got these genomic effects that we've estimated from the measured data, and we need a way to link the two. And the way we can do this is using some form of regression. Now, we can't use simple linear regression because there are more measurements than there are traits, and there are more SNPs and NIRs than there are genotypes. So traditional regression would struggle. What we can do is use one of the penalized regression methods. So either ridge regression, which effectively applies a penalty to shrink the effect sizes of the predictor variables, or Bayesian regression, which effectively puts a prior distribution that reduces the sample space. So we looked at both of these for this 2TT trial. We investigated traditional ridge regression, and we also looked at non-parametric methods based on a repeating kernel Hilbert space, or RKHS. This is quite a trendy idea from statistics recently. It's a Bayesian method that effectively specifies far fewer constraints on the problem space and lets the regression method come up with a lot of, be more flexible to match what data you've got. So we need a way to test these. And the way we did it was we used cross-validation with tenfolds. So effectively, we split the 138 genotypes into 10 roughly equally sized sets. And we fit the model based on 9 of them and predict on the 10th. And then we repeat that across all 10 groups until each one's been left out. This allows us to quantify how well each model performs, because we're now predicting based on data that the model hasn't seen, but which we know what the actual genomic effect that we measured was. It also replicates what would be required to predict for a genotype we haven't seen, where we would have the SNPs or NIRs, and we would have SNPs and NIRs data along with genomic effects for lots of other genotypes, but not for the genotypes that we haven't measured yet. And the results of these were quite interesting. So although SNPs are the traditional method that's generally been preferred for doing this, we actually found that NIRs post-filtering actually gave far better predictions. So the thing we measured was the correlation. So effectively, we got a predicted genomic effect for each genotype in the unseen set based on the SNPs and NERs and the observed data for the rest of the trial. And we effectively looked at the correlation between that and the observed BLUP for the genomic effect. And we found that NERs consistently outperformed SNPs across each of the ones we cared about. So for cellulose, both predictors did well on this. Correlations close to 1 are better, correlations close to or below 0 are bad. And we found that when we used NERs, we achieved a correlation with the observed BLUP for genomic effects of 0.82 on cellulose, and SNPs gave 0.63. For lignin, they were both a little bit lower, but again, NERs ran well ahead of SNPs, 0.67 against 0.44. And the particularly surprising result was the effect on dry matter. We found that SNPs are effectively unable to predict dry matter for unseen data, 0.07 as a correlation with the observed BLUP is very poor. That would be effectively useless for actually modeling unseen genotypes. And NERs achieved 0.52, which is still not brilliant, but pretty good. We also investigated weighted averages. So looking at taking the genomic relationship matrix that we got from the SNPs and the one we got from the NERs and averaging them to see if this improved on either of their performances individually. The best results that we achieved with that were 75% weighted towards the NERs matrix and 25% towards the SNPs matrix. This still didn't exceed the performance of NERs, it only managed to equal it. I will note here, though, one thing that's important is that Savitsky-Golay filter that we applied to the NERs. It made a big difference because when we used the unfiltered NERs that we saw were correlated, those gave predictions worse than the SNPs. They were absolutely useless. So the importance of the filtering was really on display here. Then we also, of course, compared these two different regression methods. And RKHS regression effectively gave pretty much identical performance to the ridge regression. The values for cellulose, lignin, and dry matter changed by, we're talking of the order 0.01, 0.02. And they can actually vary between runs because RKHS uses a form of random sampling. So these results wouldn't be the same every time you did them. They also vary with how you choose the tenfolds, how you split up the 138 genotypes. There was no particular evidence here to suggest that RKHS regression or ridge regression was consistently the better of the two. But since RKHS regression is more time-consuming, much more computationally expensive, we concluded that we should probably use ridge regression in practice. So that's where we got to with results, but it's entirely possible there are better ways of doing this. And we've come up with two ideas for how we might do this. Paradoxically, one is to make things more complicated, and the other is to make things simpler. The first idea is, instead of using Bayesian regression or ridge regression for the prediction step, to use a machine learning method, which increases flexibility. The obvious example for this would be random forest, which my colleague James at RSWIF is currently looking at. And perhaps at some point, if we give another talk about this, we'll have some results to show from that too. In terms of making things simpler, what we're doing at the moment by taking the whole NERS spectrum, using it to build the genomic relationship matrix, and then throwing that into our big regression method. The other idea would be to look at the things in the NERS spectrum that actually really make a difference. Try to characterize each spectrum by a few key features, the height and duration of each peak in absorbance, the locations of the lowest values as well, and try to use those to predict the genomic effects directly. So this is another idea that we're looking at, and again, perhaps in future we'll be able to tell you a bit more about that. But there is also a bigger picture I want to come back to here, which is, how can we make this as useful as possible in reality? And the big thing here is to make sure that this actually doesn't just remain a statistical method for statisticians. It really becomes useful for plant breeders who are on the front line having to deal with the reality of these things. And the idea we came up with was something called a decision engine. So the idea would be that given the location of the plant and the traits that the breeders want to optimize for, we would come back with a set of species and genotypes which would be likely to do well given those conditions based on the measurements that we have of the traits at that location for other genotypes and the measurements we have of SNPs or NERs for all of the genotypes we could consider. And we'd also at that point like to provide a sort of plausible range of forecast yields. So if we decide to grow a plant of this genotype in this location, how well do we think it could do? And this is closely linked to the other two work packages, because the model that powers Decision Engine is heavily informed by the data that we collect. And to some extent, that also goes the other way. What data we want to collect in the field could be informed by what model we've built. But also, the model is only really useful if breeders trust it. So the explainability aspect of it, which is a problem in AI in general, that AI doesn't give you clear explanations to humans of what's going on, that becomes critically important to make sure that the plant breeders know why we should go with the plants that we're suggesting. But also, it's important to take into account their existing knowledge too, because it might turn out that we can augment the model even further by correcting biases in it, errors, things that aren't captured by the SNPs and NERs. And we can only do that if we know what they know effectively. So that's where we're up to effectively. And I hope that was interesting. Thank you all for listening. And if you have any questions, I'm happy to hear them.
Great. Thank you very much, Stephen, for the excellent talk. And I'm going to open the floor to questions or comments for Stephen. Please feel free to post your comments in the chat. Till we have any questions, I have a couple of questions that I can ask. From a technical point of view, a big part of your analysis relies on this matrix A that has size n by n. What are the typical sizes of this matrix?
So the A matrix is a matrix where, so in the example we had 1,050 points in our NERs. So for each plant, we measured the absorbance of light at 1,050 different wavelengths. Mm-hmm. And then we had 138 genotypes, 552 plants, but 138 genotypes. So the matrix A that's set size n by n is effectively the average absorbance of plants of each genotype at each location. So 1,050 measurement locations, 138 genotypes, and each entry of the matrix is the average absorbance of the 4 plants of that genotype at that wavelength. And the same for SNPs, the average prevalence of that polymorphism in the genomes of the plants of that genotype.
Perfect. Is there space for model reduction or like an agent value decomposition or a singular value decomposition for reducing the size of that? Would that be necessary to understand which are the principal directions, let's say, of this matrix, assuming linearity?
So interestingly, the model that we fitted, the RKHS model, that actually does eigenvalue decomposition inside its process. So we used an R package called BGLR, which is about Bayesian generalized linear regression, but applied specifically to genomic prediction. For the ridge regression, we use another package, RRBLUP. These are packages in R, the statistical programming language. And BGLR, by default, does these eigenvalue decompositions.
Great, thank you. We have a question in the chat. Have you evaluated the model's goodness of fit, such as by calculating the AIC to determine which model performs best?
Not really. So the way we did it was by this cross-validation, just trying to compare how well the model predicted on unseen data to the values that we know we should be getting for that unseen segment of the training data. In terms of AIC, I don't know how useful that would necessarily be, because that's based on a number of model parameters. But for Bayesian regression and ridge regression, you've effectively shrunk the, you've kind of done the feature selection in that way instead, because you've shrunk a lot of the parameter, the values associated with predictors to 0, to reduce the search space to something that's manageable. I don't think, I'm going from memory here, this is something I haven't looked at in a long time, but I don't think AIC applies brilliantly well to ridge regression and Bayesian regression methods. Yes, the stuff that they do is kind of baked in already in a way.
Great, thank you. So Daniel is asking a question. I was interested that you use the BLUP in an area that, and then one of your regression models was RKHS implementation of kernel regression. Too many acronyms here. Both are the same, but are they used differently?
Yeah, so this is a really good question, actually. There's something I completely missed when I first started thinking about this analysis, is that we were actually using very similar methods in the two different steps. So the BLUP that we used was effectively for the observed genomic effect. So trying to average the genomic effect across the plants of the same genotype based on the trait measurements of each of them. We could have done that just by simple averaging, but BLUPs are slightly more robust to particularly weird one-off outlier plants. In terms of the regression step, the RKHS, I'll be honest, I mostly used default implementations. I didn't look at this too closely. I pretty much just threw things in and saw what stuck, and it seemed to do fairly well, but not so well that I'm too worried about fine-tuning it to go forward, because it only managed to equal the performance of a much simpler method. So yeah, sorry, that's probably not a very helpful answer, Daniel. Apologies for that, but I hope that was a little bit useful.
Thank you so much, Stephen. Sarah, I have Sarah Perez with me as well as a co-host. I don't know if you would like to make any comments or you want to add something.
I will actually have not a technical question, but a curiosity rather. So you have, I guess, in your measurement different individuals among a given genotype, right? So I was just curious if your approach enables to detect spontaneous DNA mutation that may be like due to like plant selection by themselves to be more resistant to any kind of disease or things like that.
That's really interesting, actually. I don't think we have looked at that. So the one thing I will say is Miscanthus is very disease resistant in the first place. It's very rare that Miscanthus plants actually die before their natural lifespan, but particularly in Britain, I think a lot of the things that they might be vulnerable to aren't really prevalent here. I suppose to some extent, you could say that by looking at the polymorphisms, we potentially capture some of that anyway, because we are actually looking at what's going on within the genome of each genotype. But yeah, that's definitely interesting. I probably need to talk to some of the more biology-focused people to see if that would be useful. But I'll make sure I do that, because that does sound really
sounds interesting, like perspective.
Yeah, thank you.
Thank you, Sarah. And join me to thank Stephen once again with a virtual clap as well. Stephen, thank you very much.
Thank you.
And we're going to move to our second speaker today, Dr. Timothée Bacri. A few words about Timothy. He's a postdoctoral research fellow at the Land Environment Economics and Policy Institute at the University of Exeter. He holds a PhD in mathematics from the University of Bergen, specializing in statistics. Currently, he contributes to the ADTRIS project, AI-elevated decision support by digital twins for restoring and enhancing ecosystem services, where his focus is on integrating various models developed by his team into a user-friendly tool designed for non-specialists aimed at assessing the impact of tree planting. Previously, Timothy's research centered on combining optimization framework template model builder with a hidden Markov model parameter estimation. Timothy, we look forward to your talk.
Thank you for the introduction. So I will talk about how we do we developed a decision support tool in the project that aims to help decision makers decide where to plant trees in a given area to maximize certain outcomes, such as the carbon stored in the ground by the trees, and while also trying to avoid planting everywhere that's possible because of, well, financial constraints. So as a quick introduction to the tool, we have three tabs currently in the tool. So the idea is that the user starts with defining a shapefile, which is a region in the UK that they are interested in, and then then the app divides that into individual parcels. Now on the screen, you can see that on the right-hand side, there are a lot of different outcomes, and with sliders, those represent targets that we want to hit. So the idea is, for example, to specify a minimum amount of tons of CO2 that we would like the planting strategy to store in the ground. We also have some biodiversity measures that we want to hit as a minimum, and we want to also minimize the area planted. And the idea is that this tab will find the optimal strategy that best corresponds to all of the sliders together. Now, the problem with this is that it only shows the optimal strategy, and some of those targets may be slightly variable. For example, we might want to increase the number of carbon stored and without caring as much about biodiversity measures. So we have a problem here in tab 2 where we show fairly different strategies that still meet the cutoffs that we defined in tab 1, but in different ways. So for example, the amount of carbon stored might be greater in the first strategy showed, but in the second one it might be lower while making the amount of pollinators larger to compensate. So the idea is here to show 4 different target compatible strategies. I am working on those tabs 1 and 2. Tab 3 will have the purpose to elicit preferences from the user, for example, to decide if carbon matters more to them than the area that is planted, or then the number of visitors that is planned to arrive in the area given a planting strategy. I am not working on that, and I will not go through this one at all, but it is part of the work that we're doing. So I will delve into Bayesian optimization and the part that I'm doing, and explain a few concepts related mainly to how it works. So the acquisition function, some data dimension or augmentation technique called random embeddings, and the end results that I can get from all of this. So as a brief reminder, the purpose of optimization, which I'm sure everyone here is familiar with, is given a random set of points or given a function, find the extremum. We'll be focusing on the minimum. Frequentist methods typically evaluate the function a lot of times and try to find the next best point that is hopefully the extremum. Now, the problem is that the objective function can be really costly. In our case, we're dealing with simulators that to compute the carbon intake, and those can take days to run. So we do not have the luxury to evaluate the function a lot of times. Also, in my case, I consider the objective function to be a black box. These are incompatible with classical optimization. So I'm looking at a Bayesian optimization. So I won't go in the details of all of this, but the idea is we start by emulating the objective function through a surrogate model. I'm using mainly GPs. And then we figure out which set of points is worthy to look at through the GP, then evaluate the objective on that new set of points and refine and improve the GP through that, and keep going until we get better and better extremum candidates. Now, for the tab 1 and 2, we'll have the work with this framework, but they have a slightly different ways. They have a slightly different way of getting the interesting results. So for tab 1, we're looking at the optimal point. So this is a classical optimization problem. For tab 2, we're instead trying to find to explore the entire space of compatible strategies. So the problem is slightly different because we do not have one extremum. We have a whole set of minimums that we want to look at. So the objective function is slightly different. For tab 1, I call it f1, and for tab 2, I call it f2. So for tab 2, the goal is to find the set of zeros of the function. So now I will explain a bit how I wrote the objective functions that I'm using here. The first one, so the p's represent the preferences of the user. Currently, I'm only using 1 as a constant because we don't have an effective way of retrieving the utility of the user. P denotes a planting strategy. Z of P is an outcome that we would want to either minimize or maximize. So the idea is, for example, on the first line, we sum the area size of each parcel, and we want to minimize the entire sum of areas because, well, we don't have unlimited money. On the second line, Z of P to maximize is meant for outcomes such as carbon, that we would like to have a strategy that maximizes the amount of carbon stored in the ground. Now, the sliders set on the first page represent targets that we want to meet at minimum or at maximum. So to do that, to represent that, I implemented penalties, very simple penalties of 100 times the difference to the targets if we are outside of those targets. So for example, the area that we want to minimize, if we go above a ceiling, we add a massive penalty to discourage this. Similarly, for the carbon, if we go below a certain floor level, the idea is to add, again, a massive linear penalty to discourage those planting strategies. So the penalties are made to, so because we want to minimize F1 and F2, the penalties are made to be positive and have a minus sign, and max or min to go in the right direction. For tab 2, the function is very similar, except we do not really care this time about maximizing the amount of carbon stored or minimizing the area planted. We only care about the target compatible space. So we only look at penalties if we are outside of the bounds that we defined in the sliders. So the sliders are, as a reminder, on the far right-hand side of this. The user can set them, and every time they set them, currently we would look at a whole new Bayesian optimization loop. So in order to do this, there were a few, I had to, because the planting strategy is very high dimensional, it has 1 dimension per parcel at least, plus what I'm not considering currently is also the year of planting, which adds a whole number of dimensions, and the type of species of trees to plant, which again keeps adding to the dimension of the inputs. So because I'm dealing with at least a few hundreds of inputs at the same, parameters at the same time, GPs do not tend to be performant on those. So I needed a way to reduce the dimension for those. These dimension reduction techniques suffer from some issues. So instead, I went with something called random embeddings. The paper that introduced it called it random embeddings for Bayesian optimization, or RAMBO for short. And I will describe that in the next slide fairly briefly. So ignore the blue box, but the idea is to start from a low dimensional space So here it would be the red line in the y with d equals 1. The idea is to generate random data in a low-dimensional space, then multiply that low-dimensional data with a random matrix, a random non-square matrix that would augment the dimension of the data. Now, and that would generate a so-called embedding space in, well, in a higher dimensional space. So the idea is to generate low-dimensional data meant for the GP and augment it to higher dimensional data that we could pass on to the objective function. Now, the original paper mainly considers high-dimensional continuous data. I have to deal with categorical data, at least currently. So the way they do this is constrain the high-dimensional, the embedding space to box constraints, and then try to hit mainly that box by doing some smart projection onto the box. And they try to find a good space in the low-dimensional, and they try to find a good subset of the low-dimensional space that best matches the box. So the main reason for doing this is that they assume that a lot of the high-dimensional parameter space is unimportant. So the idea is, for example, in 2 dimensions, if you look at the right graph, on the vertical axis is the objective function, and the idea is that we are dealing with a function where x1 makes the objective change, but x2 has almost no effect on the objective. So the idea would be to go slightly through x2, but mainly through x1, and explore mainly in the direction of x1 while trying to ignore most of x2 because it doesn't have an effect. And the idea is this in more than 1 and 2 dimensions, that most of the high dimensional space is completely irrelevant and we can avoid generating data from there, but instead mainly go through the important dimensions. Now, another part of the framework I'm using depends a lot on the choice of acquisition function, and there is some amount of work to do there. Currently, I'm using what's called the expected improvement. So the main idea is that the improvement is defined as how much better is the new point compared to the old one, or 0 if it's worse. And the idea for expected improvement is to approximate this with the Gaussian and get the parameters of the approximation through the GP. Now, there is a way to make this slightly better, because for tab 1, this works as it is without issues. But for tab 2, I know that the minimum of the objective is 0. And by default, this acquisition function will try to go below, which is a problem because there is no below for the second tab. 0 is the global minimum, and we try to find all the values that correspond to it. But so I'm sure you're all familiar with this, but so this is an example of what the acquisition function would look like given a GP trained on data represented in green. Blue corresponds to the mean predictions of the GP, and in red is plus or minus the standard deviation. And the idea is that when the variance is high, but the prediction is also high, the expected improvement increases, or when the predicted mean is low, but also then the acquisition function also increases. So we try to find the most likely next point by maximizing the acquisition function. So there is some work in making the acquisition function be aware that the minimum is known, is 0, so that it doesn't give those minimums priority for no reason. I have not implemented that yet, but it's I'm going to be looking into that in the next few days or weeks. That's about it. Sorry. As GP kernels, I have tried a few. So far, the main ones that seem to work are the Matern and squared exponential. I had tried the diffusion kernel on the higher dimensional space, but it didn't perform much better, and in fact was a lot slower because we were dealing with a very high dimensional space. So I'm mainly using the Matern one. Squared exponential is also good, but not that much better. So for tab 1, this is a brief summary in two dimension with the sum of areas and sum of carbon stored that I'm searching for. And I'm searching for the value that best for the planting strategy that maximizes or minimizes the sum of area or sum of carbon. And that line makes sense because, well, if I plant in more places, I both increase the area and the carbon stored in the ground. So these are all the strategies that are found when looking to find the optimal one. So the red lines represent the thresholds set by the users, so the sliders. So the idea is we're mostly interested in the in the top left corner. So if I zoom in, those are the strategies that I find. And for tab 1, I'm looking for the best one among those with respect to how important is carbon versus how important is area. So here, I'm not doing any rescaling. So the one that maximizes the carbon sum would be the optimum. So for example, if I decide to do a loop of 10 iterations, it takes me roughly 1 minute to search through approximately 40,000 strategies. And for the town of Falmouth, this is the best strategy that I find. By itself, it's not very interesting, but well, we see that the total area target and the total carbon targets are met. We find a strategy that best goes in above the carbon and below the area in about 1 minute. Tab 2 is very similar. We still get a large space of possible strategies, and we're also looking at the top left hand corner for the target compatible ones. And this is a map I made of this that shows the space is basically a histogram of the parcels where each parcel color represents how often a parcel was planted among all the target compatible strategies. There is some room for improvement, but so far it seems to work decently well in a reasonable amount of time. Each time a user would move a slider, it would take roughly 1 minute to have a decent search through the space of possibilities. And that's the end of my talk. Oh, yeah, sorry. The short-term work will be to look into maybe other kernels. Multi-objective optimization is something I need to look further into. So far, I'm doing very basic one with no special multipliers. Similarly, trying to optimize the acquisition function with the multi-start might be something interesting. Longer term, I'll need to integrate everything else that the other people I work with are doing, such as allow planting other tree species, planting times, have crop yields as an outcome measure, and other things like that. And now that's really the end of my talk.
Great. Thank you very much, Timothée, for the excellent talk. And please post any questions you have for Timothée in the chat. Sarah, do you have any questions or comments?
Yeah, I will have a comments question. So I'm not sure to get what you want to do at the end. Like, do you want to, in the case of multi-target objective, right? You want to minimize both the targets and reach somehow the Pareto front, or do you still allow you to have like biased target, meaning that one would be prevalent with respect to the other?
So I'll go back to the definition to how I define the objectives. So for tab 2, the idea is to only have only care about target compatibility, but still integrate some user preferences in the P's here would represent the user preferences, but we are not looking to maximize or minimize outcomes. We're only looking at all the possible strategies.
All right. Okay. But don't you want to still having like a small planting area and a maximum carbon storage at the end? So yeah,
Not
I'm just
really.
confused about getting
the
like the user.
The idea here for the user is to present alternative options that are not optimal, but might be. So the idea here would be to do some sort of clustering to find, clusters of very similar but different strategies that meet the targets, but in different ways. So for example, they might have preferences that they cannot express for, I don't know, they want tree carbon and pollinators to be increased, but they don't know exactly how much. And this tab would allow them to go through the clusters and see different ways of achieving their goals. For example, something that we have talked about a little bit, but aren't moving much further with is connectedness. So for example, it might be that they want to plant in a few close locations, a lot of parcels in one location, and instead of having everything widespread, that would be, I don't see currently a way of doing that easily. And so this going through a lot of alternatives would allow them to see different options without being able to without being able to quantify them.
All right. Thank you. Thank you for clarifying. Very interesting.
Thank you, Sarah. We don't have any other comments in the chat. So join me to thank Timothy and Stephen once again for their very insightful talks. If there are any other questions, please feel free to directly contact Timothy and Stephen and discuss further about their work. So thank you both for your talks. Some closing remarks. We're very excited to announce that the next week on Tuesday, 24th of September, we will launch the next phase of AI for Net Zero webinar series. So now we're going to open it to external speakers as well on top of UKRI AI for Net Zero projects. So our first external speaker will be Dr. Kremer, co-director of the TU Delft AI Energy Lab, who will present weekly supervised graph neural networks for distribution system state estimation. Also, I'm very, very glad to announce the following event that we will be organizing among all the UKRI AI for Net Zero projects. This will be the first conference on AI for Net Zero, accelerating the net transition with AI. This will take place 16th to 19th of September 2024 at the University of Exeter. It will be a 4-day event, bringing together leading experts and high-profile speakers to discuss how AI can drive meaningful progress towards achieving net zero. So we will have a variety of presentations, panel discussions, breakout sessions, talks, hackathons, and poster sessions. And at the moment, we're accepting abstracts for talks and posters. There should be a link posted in the chat, and please feel free to follow that link to find out more about the conference. I'm looking forward to seeing everyone at Exeter in December as well in person. Great. With that, I would like to thank everyone once again.
Thanks for inviting.